information flow from DNA to RNA to protein. Let us say we want to make a building in IIT campus, which is in BOMBAY area of Mumbai. So, the left side shows the map which shows that where this area is. So, DNA is doing something similar type of function. DNA is the genetic blueprint which contains only the information. Now, once the site is decided, then map has to be created for the building. So, RNA is the molecular photocopy which can be used on the site from the cell contractors. Now, the building has to be prepared. To do that, something similar in the body is proteins, which are the building material. Now, you need mortar and different type of bricks to make the building, and similar type of function being performed from the proteins in body. So, if you look at this way, DNA is providing the genetic blueprint, RNA is providing the molecular photocopy, and then proteins are providing the building material. So, proteins can provide and transform the one dimensional information from the sequence to the three dimensional functional information. This orderly and unidirectional flow of information, which is encoded in the base sequence of cells from DNA to RNA and protein, that is known as central dogma. Although now there are many evidences that challenges the linear logic of central dogma, but based on the premises that DNA encodes mRNA and mRNA encodes the protein, one cannot deny the conclusion that genes are the blueprint for the life and proteins are effector molecules. Due to this fact, the central dogma has guided research at the systems level. Before we move on to discussing about the structure and function of DNA, let me take you to some of the historical perspective, the milestone discoveries which are related to the DNA. So, first one is Mendel's law of genetics in 1865. Mendel gave very fundamental laws of genetics. The discrete factors, which are now known as genes, can transmit characteristics from one generation to the next generation. So, diploid individual must contain two copies of genes. Each parent can transmit one copy to the next generation. After this hereditary laws from Mendel, lot of research started in this area, and then one of the major milestone was DNA double helix structure, which was discovered by Watson and Crick in 1953. Watson and Crick published a paper in 1953 in Nature, and they described, we wish to suggest a structure for the salt of deoxyribonucleic acid DNA. This structure has novel features which are of considerable biological interest. From that time, the structure and function of DNA has been a subject of great research interest in the field of biology. So, shown in the picture is James Watson, and I had an opportunity to meet with him in Cold Spring Harbor. So, I have shown that picture here. Now, in 1966, Nierenberg, Khorana and Hawley, they determined the genetic code. Another major milestone discovery was recombinant DNA technology, which was developed in 1972 by Cohen and Boyer. In 1977, the DNA sequencing methods were provided by Sanger, Maxson and Gilbert. Now, let us move to the 1990s. One of the major interesting area of research in biology was cloning. So, cloning is producing a cell or organism with the same nuclear material as another cell or organism. Dr. Ian Valmont of Roslin Institute and his colleagues cloned a sheep, Dolly, which was first large cloned animal from the somatic cells. During this time, the Human Genome Project was initiated, and many of the genome projects and especially the Human Genome Project was completed in 2003, which was an initiative from International Human Genome Sequencing Consortium as well as Celera Genomics. And now very recently, different type of next generation sequencing approaches have made sequencing so much fast and affordable that it is revolutionizing the biological research at the genomic level. So, after giving you the perspective, some of the historical background, let us go to some of the basics of DNA, RNA and proteins. Although this is not the directly linked to the proteomics, but to understand the concept at the systems level, I think it is very important to refresh your fundamentals of DNA, RNA and proteins. So, deoxyribonucleic acid or DNA, which stores and transmits all the genetic information, is a long polymer of nucleotide monomers that assumes a complex double helical structure. So, main function of nucleic acid is the storage and transmission of genetic information, and there are two major classes, DNA and RNA. Now, nucleoside is when you have a sugar and base. When phosphate molecule is also added, that gives